Good evening, my dear students. Welcome to Sankalp Learning Classes. Already we have started the chapter Principles of Inheritance and Variation, right? So already we have completed the two classes on this chapter. So today this is the last part we are going to study on this chapter. Okay. Yes, what we learned in the last class? Yes, very good evening to all. Yes. So in the last class, we studied about the sex determination, right? So in case of grasshopper, so there is a XX and X0 type of the sex determination, where the females, they are having the two X chromosomes and males, they are having the X0, means only one X chromosome. So that is the sex determination in grasshopper. Then sex determination in mammals, very important, that is XX and XY type of the sex determination, both mammals and the Drosophila, where the two X chromosomes are present in females along with the autosomes, 22 autosomes plus XX chromosomes plus 22 autosomes plus XY chromosomes in male. So that is the sex determination in mammals, mainly in the human beings. Okay, sex determination in human beings. Then sex determination in birds, that is ZW type of sex determination, where females are showing the heterogamety. So you know the meaning of heterogamety. Females are showing this heterogamety where Z and W type of the gametes, so that is present in the female egg, and ZZ type of the uh, chromosomes, ZZ type of sex determination done in the males. Okay, here males are homogametes. So that we already studied in the last class. So next thing is very important, sex determination in honeybee. So here sex determination in honeybee is based on the number of sets of chromosomes an individual receives. Okay. Here, an offspring formed from fertilization of sperm and egg developed into either queen or a worker female. Just see here. Here, the sex determination in honeybee, it is called as haplodiploid type of the haplodiploid type of the sex determination. Here, if the egg from the female fuses with the sperm, so resulting offspring, so the new offspring that will develop into a female queen or worker. Okay, female worker. Here the fusion of egg and sperm that leads to the formation of the female queen or worker, that is called as the that is also a female. Okay. If the unfertilized egg develops as a male by means of parthenogenesis without fusion of male and female gamete, here just imagine egg and the sperm. Okay. If this egg fertilizes directly, then it will develop into a male honeybee. Okay. So without fertilization, then here the males are haploid or diploid. So just comment, write down the comment. So here the male honeybees are haploid or diploid. Yes, very good. They are haploid. Alva? So they are haploid. Haploid ಆಗಿರುತ್ತೆ because without fertilization, ಅಲ್ಲಿ fertilization ಆದ್ರೆ ಮಾತ್ರ ಏನಾಗಿರುತ್ತೆ, ಡಿಪ್ಲಾಯ್ಡ್ ಆಗುತ್ತೆ ಅಲ್ವಾ. ಸೊ ದೇ ಡೆವಲಪ್ ಫ್ರಮ್ ದಿ ಗ್ಯಾಮೆಟ್, ದಟ್ ಈಸ್ ಎಗ್ ಇಂದಾನೆ ಡೈರೆಕ್ಟ್ ಆಗಿ ಡೆವಲಪ್ ಆಗುತ್ತೆ ಮೇಲ್ಸು. ಸೊ ದೇ ಆರ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ಸ್. The male have half number of chromosomes than that of the female. The female are diploid having the 32 chromosomes. Females are diploid and males are haploid, that is having the 16 chromosome number of chromosomes. So this is called as haplodiploid sex determination system. So this is the haplodiploid sex determination system. Here you can understand this process. This is the diploid female. So it will undergo the meiosis and form the eggs. Okay. So eggs without fertilization. If these eggs without fertilization, eggs, they will not undergo fertilization, they develop into a haploid males. So egg which is having the 16 chromosomes and these males also having the 16 chromosomes. ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? ನೆಕ್ಸ್ಟ್ ನೋಡಿ, ಈ ಡಿಪ್ಲಾಯ್ಡ್ ಫಿಮೇಲ್ ಏನ್ ಮಾಡ್ತಾ ಇದೆ, ಎಗ್ ಪ್ರೊಡಕ್ಷನ್ ಮಾಡ್ತಾ ಇದೆ. ಅಂಡ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಮೇಲ್ ಅಂಡ್ ದೀಸ್ ಮೇಲ್ಸ್, ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಮೇಲ್ಸ್ ಪ್ರೊಡ್ಯೂಸಿಂಗ್ ದ ಸ್ಪರ್ಮ್ ಪ್ರೊಡಕ್ಷನ್ ಮಾಡ್ತಾ ಇದೆ. ಎರಡು ಫರ್ಟಿಲೈಸೇಷನ್ ಆಯ್ತು. ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗಿ ಸೊ ವಾಟ್ ದೇ ವಿಲ್ grow into, they develop into a queen or worker female. So they develop into a female. Okay, so this type of the sex determination is called as haplodiploid type of the sex determination. So very important for your exam, very important for your exam, that is haplodiploid type of the sex determination in honeybees. And here the male honeybees, they are called as drone, and female, so either they may be queen or the worker bees. Okay, so this is about the sex determination in different organisms. So we have studied sex determination in birds, then Drosophila, mammals, and the grasshopper, right? So these are the different in different organisms, the sex determination method in the different organisms. So let us study about the next topic, mutation. What is mutation? You should know the meaning of the mutation. Here, mutation is a phenomenon which results in alteration of DNA sequences and subsequently result in changes in the genotype and the phenotype of the organism. In addition to recombination, mutation is another phenomenon that leads to the variation in the DNA. Okay, so this mutation is the phenomenon. Just imagine this is the DNA. Okay, so this is the DNA. ಇದೊಂದು DNA ಅಂತ ಅನ್ಕೊಂಡ್ರೆ, ಮ್ಯುಟೇಷನ್ ಅಂದ್ರೆ ಏನು ಅಂದ್ರೆ, ಸೊ ಅಲ್ಟರೇಷನ್ ಆಫ್ DNA ಸೀಕ್ವೆನ್ಸಸ್ ಕಾನ್ಸಿಕ್ವೆಂಟ್ಲಿ ರಿಸಲ್ಟ್ ಇನ್ ದ ಚೇಂಜಸ್ ಇನ್ ದ ಜಿನೋಟೈಪ್ ಅಂಡ್ ದ ಫಿನೋಟೈಪ್. ಇಲ್ಲಿ ನೋಡಿ, ಈ ಒಂದು ಬೇಸ್ ಪೇರ್ ಏನಾಗಿದೆ, DNA ಅಲ್ಲಿ ಚೇಂಜ್ ಆಗಿದೆ. ಇದೇನಾಗಿದೆ, ಚೇಂಜ್ ಆಗಿದೆ. ಇದು ಚೇಂಜ್ ಆಗಿರೋದ್ರಿಂದ, DNA ಅಲ್ಲಿ ಚೇಂಜ್ ಆಗಿರೋದ್ರಿಂದ ಏನಾಗಿದೆ, ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಕೂಡ ಏನಾಗಿದೆ, ಚೇಂಜ್ ಆಗಿದೆ. ಸೊ ದಟ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ ದ ಮ್ಯುಟೇಷನ್. ಓಕೆ, here loss or the deletion or gain or insertion or duplication of segment of DNA result in the alteration of chromosomes. ಇಲ್ಲಿ ನೋಡಿ, ಒಂದು DNAಗೆ, ಇದೊಂದು DNA ಅಂತ ಅನ್ಕೊಂಡ್ರೆ, ಓಕೆ, ಈ DNA ಒಂದು ಎಕ್ಸ್ಟ್ರಾ ಎಡಿಷನ್ ಆದ್ರೂ ಕೂಡ ಏನಾಗತ್ತೆ, ದರ್ ಇಸ್ ಎ ಅಬ್ನಾರ್ಮಲಿಟಿ. ಆರ್ ಒಂದು ಡಿಲೀಟ್ ಆದ್ರೂ ಕೂಡ, ಒಂದು ಸ್ಮಾಲ್ ಪೀಸ್, ಒಂದು ಸ್ಮಾಲ್ ಚೇಂಜಸ್ ಆದ್ರೂ ಕೂಡ, ಇದೊಂದು DNA ಅಲ್ಲಿ ಇಲ್ಲಿ ಒಂದು ಸ್ಮಾಲ್ ಚೇಂಜಸ್ ಆದ್ರೂ ಕೂಡ ಏನಾಗತ್ತೆ, ದಟ್ ರಿಸಲ್ಟ್ಸ್ ಇನ್ ಅಬ್ನಾರ್ಮಲಿಟೀಸ್ ಆರ್ ಅಬರೇಷನ್. Chromosomal aberrations are commonly observed in cancerous cells. Mutation also arise due to the change in the single base pair of DNA. This is known as the point mutation. ಇಲ್ಲಿ ನೋಡಿ, so just imagine this is the DNA. ಡಬಲ್ ಸ್ಟ್ಯಾಂಡ್ ಇರುತ್ತೆ ಅಲ್ವಾ, DNA ಅಲ್ಲಿ ಡಬಲ್ ಸ್ಟ್ಯಾಂಡ್ ಇರುತ್ತೆ. ದೆನ್ ಇಲ್ಲಿ ಬೇಸ್ ಪೇರ್ಸ್ ಇರುತ್ತೆ, ಅಂದ್ರೆ ಅಡಿನೈನ್ ATCG ಅಂತ ಈ ರೀತಿ ಬೇಸ್ ಪೇರ್ಸ್ ಇರುತ್ತೆ. ಓಕೆ, ATCG ಈ ತರ ಏನಿರುತ್ತೆ ಬೇಸ್ ಪೇರ್ಸ್ ಇರುತ್ತೆ. ಹೀಗೆ ಇರಬೇಕು ಅಂತ ಇಲ್ಲ, ಬೇರೆ ಬೇರೆ ತರದಲ್ಲೂ ಇರ್ಬೋದು. ಆದ್ರೆ T ಜೊತೆ ಬೈಂಡ್ ಆಗಿರುತ್ತೆ ಹೈಡ್ರೋಜನ್ ಬಾಂಡ್ ಜೊತೆ, ಅಂಡ್ G ಜೊತೆ ಬಾಂಡ್ ಆಗಿರುತ್ತೆ. ಇಫ್ mutation also arise due to the change in the single base pair of DNA. ಇದನ್ನ ಒಂದು ಬೇಸ್ ಪೇರ್ ಅಂತ ಕರೀತಾರೆ. ಈ ಒಂದು ಸಿಂಗಲ್ ಬೇಸ್ ಪೇರ್ ಚೇಂಜಸ್ ಆದ್ರೆ ಇಲ್ಲಿ AT ಇರಬೇಕಿತ್ತು ಅಲ್ವಾ, AG ಇದ್ರೂ ಕೂಡ ಏನಾಗತ್ತೆ, ಸೊ ದಟ್ ಈಸ್ ದ ಚೇಂಜ್. ಸೊ ದಟ್ ಆಲ್ಸೋ ಕಾಸ್ ದ ಮ್ಯೂಟೇಶನ್. ಸಿಂಗಲ್ ಬೇಸ್ ಆಫ್ pair of DNA, this is known as point mutation. ಸೊ ಇದನ್ನ ಪಾಯಿಂಟ್ ಮ್ಯೂಟೇಶನ್ ಅಂತ ಕರಿತೇವೆ. ಸಿಂಗಲ್ ಬೇಸ್ ಪೇರ್ ಚೇಂಜ್ ಆದ್ರೆ ಇಲ್ಲಿರುವಂತದ್ದು ನೋಡಿ AT ಈ ತರ ಇರ್ಬೇಕಿತ್ತು, ಇಲ್ಲೇನಾಗಿದೆ AG ಇದು ಬಿಟ್ಟಿದೆ. ಸಿಂಗಲ್ ಬೇಸ್ ಪೇರ್ ಚೇಂಜ್ ಆದ್ರೂ ಕೂಡ, ಸೊ that is also called a mutation, that is called as the point mutation. Deletion and insertion of base pairs of DNA causes the frame shift mutation. ಇಲ್ಲಿ ನೋಡಿ, ಈಗ DNA ಅಲ್ಲಿ ಏನಾಗಿದೆ, ATCG ಇಷ್ಟೇ ಇರ್ಬೇಕಿತ್ತು. ಇಲ್ಲಿ ಏನಾಗಿದೆ, ಒಂದು ಎಕ್ಸ್ಟ್ರಾ ಆಡ್ ಆಗಿದೆ, ಎಕ್ಸ್ಟ್ರಾ CG ಆಡ್ ಆಗಿದೆ. ಸೊ ದಿಸ್ ಇಸ್ ಎಕ್ಸ್ಟ್ರಾ. ಓಕೆ, ಎಕ್ಸ್ಟ್ರಾ ಆಡ್ ಆಗಿದೆ ಅಥವಾ ಏನಾಗಿದೆ, ಡಿಲೀಟ್ ಆಗಿದೆ. ಒಂದು ಡಿಲೀಟ್ ಆದ್ರೆ ಅಥವಾ ಎಕ್ಸ್ಟ್ರಾ ಆಡ್ ಆದ್ರೂ ಕೂಡ ಏನಾಗುತ್ತೆ, ಸೊ ದಟ್ ಈಸ್ ಆಲ್ಸೋ mutation. Deletion and insertion of base pairs of DNA causes the frame shift mutation. Addition or deletion of the single base pairs of the DNA that cause the frame shift mutations. Okay. Here, what are the causes for the mutation? Mutation ಆಗ್ಲಿಕ್ಕೆ reasons ಏನು? Mutations can be caused by different agents known as mutagens. Mutation cause ಮಾಡುವಂತ agents ಗೆ mutagens ಅಂತ ಕರೀತಾರೆ. Mutagens can be physical or chemical. ಸೊ ಅದು chemical mutagens ಇರಬಹುದು, physical mutagens ಇರಬಹುದು. Physical mutagens ಅಂದ್ರೆ like temperature, radiation, UV rays, alpha, beta and gamma rays, X rays ಏನ್ ಮಾಡಬಹುದು, DNA ಅಲ್ಲಿ changes ಅನ್ನ cause ಮಾಡಬಹುದು. So that also leads to the mutation. And chemical mutagens like mustard gas, nitrous gas, so these are called as the chemical mutagens. Again, there is a type of mutation, already we have studied this. Point mutation, that is the gene mutation. So it arises due to a change in the structure of gene. This type of mutation normally alters the information conveyed by the gene. It alters its message. In this, there is a mutation is in only one nitrogenous base of the triple codon and thus known as the point mutation. So that is the point mutation. Frame shift mutation ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, addition or loss of more than one nitrogenous base. It is result in the shift of nitrogenous bases and reading of the codon affected. Hence the protein formed different or may not be formed at all. So frame shift mutation ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂತ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೆ ಅಲ್ವಾ ನಾನು ಲಾಸ್ಟ್ ಅಲ್ಲಿ. ಸೊ ನೈಟ್ರೋಜನಸ್ ಬೇಸ್ ಪೇರ್ಸ್ ಏನಾಗುತ್ತೆ, ಆಡ್ ಆಗುತ್ತೆ, ಮೋರ್ ದನ್ ಒನ್ ಅಥವಾ ಡಿಲೀಟ್ ಆಗುತ್ತೆ. ಪಾಯಿಂಟ್ ಮ್ಯುಟೇಷನ್ ಅಲ್ಲಿ ಸಿಂಗಲ್ ಬೇಸ್ ಪೇರ್ ಏನಾಗುತ್ತೆ, ಚೇಂಜ್ ಆಗುತ್ತೆ. ದಟ್ ಲೀಡ್ಸ್ ಟು ದ ಮ್ಯುಟೇಷನ್, ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ದ ಪಾಯಿಂಟ್ ಮ್ಯುಟೇಷನ್ ಅಂತ ಕರೀತಾರೆ. Pedigree analysis. Yes, what is this pedigree analysis? Pedigree analysis, an analysis of serial records occurrence of a trait in a several generation of a human family. So here pedigree analysis, so it is an analysis of serial records of the occurrence of trait in the several generation called the pedigree analysis. So we will come to know in the next one. The first thing in this analysis to collect the family history regarding the trait of the particular. To form a pedigree chart, you should know some symbols. Okay. Here, to construct the pedigree chart, you should collect the family history as well as you can draw the that family tree. Okay, so to draw that family tree, so you should know some symbols. For males, you should use square like this. Okay, for females, you should use the circle. And if the sex is unspecified, so we don't know is it male or female, then we should use this symbol. If affected, if any person is suffering from the disease, so it is represented in the red color. Okay, if male is suffering, red color square is used. Female, then circle. And the if the sex is unspecified, that is in this symbol. Okay, then if mating means if the male and female they are married. Okay, married, so that is shown like this. So that is square and the circle joined by a line, that is the mating. Mating between the relatives means that is contagious mating, so there is a chance of disease, so that is shown in the double line. Parents above the children below in order to birth left to right. Okay, parents above and children below, so these are the parents and below these are children. Okay, so here this represents the parents with affected children, and here five unaffected offspring. So these symbols are very important to construct the family to construct the pedigree chart. Okay, ಇಲ್ಲಿ ನೋಡಿ, ಇಲ್ಲಿ ಪೆಡಿಗ್ರಿ ಚಾರ್ಟ್ ಅನ್ನ ಹೇಗೆ ಕನ್ಸ್ಟ್ರಕ್ಟ್ ಮಾಡೋದು. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ವಿ ನೋ ದ್ಯಾಟ್ ಗ್ರ್ಯಾಂಡ್ ಪೇರೆಂಟ್ಸ್. ಓಕೆ, ಸೊ ಅವರ್ ಗ್ರ್ಯಾಂಡ್ ಪೇರೆಂಟ್ಸ್, ಜಸ್ಟ್ ಇಮ್ಯಾಜಿನ್ ಗ್ರ್ಯಾಂಡ್ ಪೇರೆಂಟ್ಸ್, ಸೊ ದೇ ಆರ್ ಮ್ಯಾರಿಡ್. ಓಕೆ, grandfather and the grandmother, they have the 3 children. So just imagine grandfather, grandmother, and they have the 4 children. In that, 2 males you have to draw like this, and 2 females. Okay, 2 females like this. So here one of them is married and they have again 2 children, both are boys or males, like this. So like this you have to draw the family chart. Okay, so here we can analyze the diseases. By convention, the male members of the family are shown by the squares and females by the circles. Here if you collect the family history, so you can analyze the diseases, or if any person is suffering from the disease, so how it is transferring to the next generation. So these all things we can analyze using the pedigree chart. Okay. Yes, good evening everyone. Mendelian disorders. So there are mainly 2 types of the disorders. So what are these 2 types of the disorders? The first one is Mendelian disorders. Mendelian disorders. And second one is chromosomal disorders. Mendelian disorders and the chromosomal disorders. So Mendelian disorders, what is the meaning of Mendelian disorders? Here Mendelian disorders means which are caused by change in the single gene present on the chromosome. Okay, so this Mendelian disorder which are caused by ಚೇಂಜ್ ಇನ್ ದ ಸಿಂಗಲ್ ಜೀನ್. ಸೊ ಯಾವುದೋ ಒಂದು ಜೀನ್ ಏನಾಗಿದೆ ಚೇಂಜ್ ಆಗಿರೋದ್ರಿಂದ ಸೊ ದಟ್ ಡಿಸೀಸ್ ಈಸ್ ಕಾಸ್ಡ್. ಓಕೆ, ಸೊ ದಿಸ್ ಟೈಪ್ ಆಫ್ ಡಿಸೀಸಸ್ ಆರ್ ಕಾಲ್ಡ್ ಆಸ್ ಮೆಂಡಲಿಯನ್ ಡಿಸಾರ್ಡರ್ಸ್. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಸಿಕಲ್ ಸೆಲ್ ಅನಿಮಿಯಾ and color blindness. So these all are example for Mendelian disorders. And one more type is the chromosomal disorders. So what do you mean by chromosomal disorders? Do you know? Anybody? If you know what you mean by chromosomal disorders, Mendelian disorders means the disorder which is caused due to the change in the single gene present on the single gene, or a uh, change in the genes present on the chromosome. What do you mean by chromosomal disorder? What do you mean by chromosomal disorder? Anybody? Please write down in the comment box, those who know. Chromosomal disorder means which is the disorder caused due to the change in the chromosome number. Yes, 46 is the total number of chromosomes present in the human beings. If the chromosomal number changed, if it is 47 or 45, so that leads to the disorder, that is the chromosomal disorder. Chromosome changes in the number of chromosome that leads to the disorder, that is nothing but the chromosomal disorder. Okay, so let us study about the Mendelian disorders. In that, first one, sickle cell anemia. Okay, so example for the Mendelian disorder, that is the sickle cell anemia. So what is this sickle cell anemia? Anybody? Sickle cell anemia. So ಹೇಗೆ cause ಆಗುತ್ತೆ sickle cell anemia? It is an autosomal recessive disease. What is the meaning of autosomal recessive disease? Autosomal recessive disease andre ಏನು? Autosomal recessive disease means, autosome means which is other than the sex chromosome. 22 chromosomes plus XX or XY chromosomes will be there in human beings. These XX and XY chromosomes, they are called as sex chromosomes. So these are sex chromosomes, and other than this 22 pair chromosomes, they are called as autosomes, right? Other than this, so the chromosomes are called as autosomes. So this is sickle cell anemia is autosomal, means the genes which are present on the autosome that is changed. Okay, autosomal recessive disorder. What do you mean by recessive disorder? Recessive disorder means in the heterozygous condition. For example, okay, so the gene HBA present on the HBA gene responsible for the formation of the hemoglobin. But if any one gene is defected, HBA is correct, but this gene is defected, then the disease will not caused. Okay, ಈಗ ಒಂದು gene ಡ್ಯಾಮೇಜ್ ಆದ್ರೆ ಎರಡು ಒಂದು ಜೀನ್ ಡ್ಯಾಮೇಜ್ ಆದ್ರೆ ಏನಾಗೋದಿಲ್ಲ, ಡಿಸೀಸ್ ಕಾಸ್ ಆಗೋದಿಲ್ಲ. ಸೊ ದಟ್ ಈಸ್ ದ autosomal recessive disorder. In this, the glutamic acid in the 6th position in the globin chain is replaced by the valine. It is caused due to the mutation of this. Okay, in this disorder the red blood cells become sickle shaped, so it is called as the sickle cell anemia. Red blood cells, they become the sickle cells. The genotype of the healthy individual, so the healthy individual having the HBA genes, but while the affected is having the HBS affected ಆದ್ರೆ ಏನಾದ್ರೆ ಡಿಸೀಸ್ ಕಾಸ್ ಆದ್ರೆ ಯಾವ್ದಿರತ್ತೆ, HBS genotype will be there. Homozygous individual for the disease die early due to the destruction of the red blood cells. The individual who is heterozygous, that is HBS is the carrier. ಈ HbA HbS is the carrier because it transfer to the next generation ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗತ್ತೆ. So this HbA HbS condition, that is a heterozygous condition. Let us see how this disease is caused. Yes, okay. Here normal gene, so normal HbA gene, okay, so that is coding CTC, okay. So that is on the mRNA, so that gene DNA, that is converting into the RNA, okay, RNA. So that is GAG, that codes GAG, HbA gene, so that codes GAG on mRNA, and GAG that codes for glutamine, okay. But here in the sickle cell anemia, what happens? So the mRNA that codes for GUG, GUG ge code madutte, that GUG means that is the valine. The substitution of the amino acid in the globin protein results due to the single base substitution at the 6th codon of beta globin gene from GAG to the GUG. So ಇಲ್ಲಿ ಏನಾಗ್ತಾ ಇದೆ ಅಂದ್ರೆ hemoglobin chain, that is the peptide chain alva, so that is the amino acid chain. Here the small only one amino acid is changed in this peptide chain. Only one amino acid change agide, ಅಲ್ಲಿ ಏನಾಗಿದೆ glutamic acid ಏನಾಗಿದೆ valine ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗಿದೆ ಅಲ್ವಾ. So this leads to the mutant hemoglobin molecule undergoes the polymerization under the low oxygen tension, causing the change in the shape of the RBC from biconcave to the elongated disc-like. So that leads the sickle cell anemia. Here. HbA parental. So there is an example given. HbA HbS means carrier, okay. HbA HbS carrier parents. So when these two are crossed, so there is a chance of HbA HbA and HbA HbS and HbA HbS, right. So this yes, here you can see HbA 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 HbS and HbA HbS HbA HbS. So here only healthy one individual is there and carrier 2 and anemic, sickle cell anemic, that is HbS HbS, okay. Next Mendelian disorder, that is the thalassemia. So what is this disease? This is also autosome linked. Already I have explained. Autosome linked means recessive blood disease transmitted from parents to the offspring when the both parents are unaffected carrier for the gene heterozygous. Here this is autosome linked recessive blood disease. Thalassemia also a blood disease. If both the parents are unaffected, but if they are carrier, the children will be affected. The defect could be due to the either mutation or deletion of which ultimately results reduced rate of synthesis of one of the globin chain. There are two hemoglobin chain, that is alpha and beta chains that make up the hemoglobin, okay. So there is a reduced rate of synthesis of the globin chain that affect the hemoglobin formation. This causes the formation of abnormal hemoglobin molecule resulting into a anemia, which is the characteristic feature of disease, this thalassemia. So which result to the anemia, which forms the abnormal hemoglobin molecules, okay. And thalassemia can be classified according to which of the chain molecule is affected. In alpha thalassemia, In alpha thalassemia, production of alpha globin chain is affected. In beta thalassemia, production of beta globin chain is affected. Okay, so there are two types of hemoglobin chains are there: alpha globin chain, beta globin chain. Alpha thalassemia is controlled by two closely linked genes, HbA1, HbA2. Okay, so these are the genes responsible for the production of the alpha globin chain. So these are present on which chromosome? 16th chromosome, and it is observed due to the mutation or deletion of one or more of the four genes. The more genes affected, the less alpha globin molecule is produced. ಈಗ ನೋಡಿ 16th chromosome, just imagine this is the 16th chromosome. 16th chromosome, okay, here which has HbA1 gene, HbA2, here also HbA1, HbA2 genes are there. So this HbA1, HbA2 genes are responsible for the production of alpha chain, alpha globin chain. Here, if one gene is defected or two gene is defected, so how many genes are defected? So that affect that much less alpha globin molecules are produced. Okay, while beta thalassemia is controlled by single gene, that is HBB on the chromosome 11. So HBB single gene is present on the 11th chromosome, each parent, and occurs due to the mutation or one or both the genes. Here beta globin chain, the HBB gene, okay, HBB, here also only one gene, HBB gene. So this HBB gene, one or if both are deleted or both are undergo the mutation, then this disease occurred. Thalassemia differs from sickle cell anemia. Sickle cell anemia is also blood disease, alva? Adu kuda blood disease. Thalassemia is also blood disease. But in that, former is the quantitative problem of synthesizing too few globin molecules, while latter is the qualitative problem of synthesizing the incorrectly functioning globin. So thalassemia, that is the quantitative. Quantity kadme agate. Alpha globin chain irbodu, beta globin chain irbodu, adra quantity kadme agate. Hemoglobin production kadme agate. Anemia cause agate alva. But sickle cell anemia dalli ಏನಾಗುತ್ತೆ, RBC sickle shape ಆಗುತ್ತೆ ಅಲ್ವಾ. So that is the qualitative problem. This is the quantitative problem. Next one, phenylketonuria. So this is also a Mendelian disorder. Okay. So this is also a Mendelian disorder. It is an error, that is the autosomal recessive, that affected the person does not produce the enzyme phenylalanine hydroxylate that converts the amino acid phenylalanine into tyrosine. Okay, so there is a enzyme that converts the phenylalanine into tyrosine. Okay, so there is a amino acid phenylalanine that is converted into tyrosine in the presence of phenylalanine hydroxylase enzyme. So this enzyme converts it into a tyrosine. Thus, phenylalanine accumulates in the body and it is converted into phenylpyruvic acid. So, in the absence of this enzyme, this phenylalanine is converted into phenylpyruvic acid. So, this enzyme is not produced due to the due to the absence of genes on the chromosomes. Okay, genes present on the chromosome. So this enzyme is not produced. Enzymes, that is nothing but the proteins, alva. So this enzyme is not produced. So phenylalanine that convert into phenylpyruvic acid. So this phenylpyruvic acid, so this causes the mental retardation, and in some cases phenylpyruvic acid is secreted out in the urine in the kidney, is not also able to absorb it. Kidney is also not able to absorb it. It causes the mental retardation. So that is the phenylketonuria. Next one, hemophilia. So this is also a disease, but sex-linked recessive disease means, sex-linked means the genes which are present on the sex chromosomes, so they are changed. The genes which are present on the XX or XY chromosomes that are affected, due to this disease is caused, hence it is called as the sex-linked recessive disorder. What is hemophilia then? Hemophilia, so where the blood does not clot in the people suffering from this disorder. Male carrying a single recessive allele is affected, while the female will be hemophilic only in the homozygous condition. ಇಲ್ಲಿ hemophiliaದಲ್ಲಿ ನೋಡಿ, hemophiliaದಲ್ಲಿ the genes which are present on the X chromosome ಅಲ್ಲಿರುವಂತಹ ಒಂದು ಜೀನ್ಸ್ ಡಿಫೆಕ್ಟ್ ಆದ್ರೆ ಏನಾಗತ್ತೆ, ಹಿಮೋಫಿಲಿಯಾ ಕಾಸ್ ಆಗತ್ತೆ. ಬಟ್ ಇನ್ ಕೇಸ್ ಆಫ್ ಮೇಲ್ ಅಲ್ಲಿ ಏನಾಗಿದೆ, ಓನ್ಲಿ ಒನ್ ಎಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ ಇದೆ. ಎಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ ಒಂದೇ ಡಿಫೆಕ್ಟ್ ಆದ್ರೆ ಏನಾಗುತ್ತೆ, ಆಫನ್ಲಿ ಮೇಲ್ಸ್ ಆರ್ ಮೋರ್ ಅಫೆಕ್ಟೆಡ್. ಮೇಲ್ಸ್ ಏನಾಗಿರುತ್ತೆ, ಮೇಲ್ಸ್ ಅಲ್ಲಿ ಏನಾಗಿರುತ್ತೆ, ಜಾಸ್ತಿ ಅಫೆಕ್ಟ್ ಆಗಿರುತ್ತೆ. ಬಿಕಾಸ್ ಒಂದೇ ಒಂದು ಎಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ ಇದೆ. ಎಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ ಇರುವಂತಹ ಒಂದು ಜೀನ್ಸ್ ಡಿಫೆಕ್ಟ್ ಆದ್ರೆ ಮೇಲ್ಸ್ ಆರ್ ಅಫೆಕ್ಟೆಡ್. ಇಫ್ females only one X chromosome is affected, another one is there, so there is a less chance of affecting the females. A female carrying a single recessive allele is a carrier. A female usually not affected, as male become hemophilic only if her father, if they may become hemophilic only if father is hemophilic and mother is carrier. The male inherit the disease from the female. So males ಗೆ disease ಹೇಗೆ ಬರುತ್ತೆ, females ಇಂದ ಬರುತ್ತೆ. Okay, so just imagine XX female and it is carrier. Okay, see, the affected, only one X chromosome is affected, then male, so X chromosome is affected, so he is affected. So when they produce the baby, here there is a chance of, so female with carrier and one healthy baby male, and there is one female is affected. If both the XX chromosomes are affected, then the female will be affected, and one affected male. Here there is a chance of one affected male. Nodi, male one effect agide alva, and female, then there is one carrier female. Okay, so this is how hemophilia transfer from one generation to the next generation. See here, there is an example. Parental type, here XX chromosome which is not affected, but here hemophilic father. XH means hemophilic father. Okay, so they produce the gametes XX and XH and the Y, the sperm which is carrying the gametes. So here there is a chance of XX and XH and XY. So when you write down in the Punnett square, X and one X is affected means she is the carrier. Okay, she is also carrier. XY, so healthy male and XY, another healthy male is possible. In case of female is only one X chromosome is affected. Only female is the carrier. XHX crossed with the XY. So what will be the resulting offspring that you can see here. ಇಲ್ಲಿ ನೀವು ನೋಡಬಹುದು ಯಾವ ತರ ಆಗತ್ತೆ ಅಂತ. Next one, color blindness. What is this disease? Color blindness. It is also a sex-linked recessive disorder. Okay, color blindness is also a sex-linked recessive disorder. In this disorder, the person cannot perceive red and green light. So the person not able to distinguish the red and green color, sometimes blue color also. It shows the same crisscross pattern of inheritance. Female should be homozygous recessive for the disorder to be expressed, while the heterozygous male would be affected. So, sex-linked recessive disorder, just imagine here the female, only one X chromosome is affected, then she is called as the carrier. Okay, she is called as the carrier. In male, if the one X chromosome is affected, another one is Y, he is the diseased male. Diseased male because only one X chromosome is present in the male. Here, so here X chromosome and XC chromosome gametes. Then here carried Y. So you can see the X. Here she is the carrier offspring, and here next one X. C and XC means both are affected. Okay, so here the homozygous condition for this disease, so both are affected. Then XY, so there is the healthy male and another affected male. Okay, so like this offsprings are obtained. So ಇಲ್ಲಿ ನೋಡಿ, ಇಲ್ಲಿ ಏನಾಗ್ತಾ ಇದೆ ಅಂದ್ರೆ male is the affected, healthy female. So how the offsprings will be affected. So offspring ಯಾವ ತರ affect ಆಗುತ್ತೆ ಅನ್ನೋದನ್ನ ನೀವಿಲ್ಲಿ ನೋಡಬಹುದು. So this is called as crisscross patterns of inheritance. ಯಾಕೆ crisscross pattern ಅಂತ ಯಾಕೆ ಕರೀತಾರೆ ಅಂದ್ರೆ, ಫಿಮೇಲ್ ಇಂದ ಮೇಲಿಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗುತ್ತೆ. The female, the heterozygous female that transfer the disease into the male, from male to the female. So ಅದೇನಾಗತ್ತೆ, ಫಿಮೇಲ್ ಇಂದ ಮೇಲ್ಸಿಗೆ means grandsons. Okay, grandsons ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗುತ್ತೆ, ನೆಕ್ಸ್ಟ್ ಮತ್ತೆ ಫಿಮೇಲ್ಸ್ ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗುತ್ತೆ. ಸೊ ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ದ ಕ್ರಿಸ್ ಕ್ರಾಸ್ ಪ್ಯಾಟರ್ನ್ ಆಫ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಅಂತ ಕರೀತಾರೆ ಅದನ್ನ. Here you can see the another example. Here XC means color blind. X means she is the carrier, and the male XY, healthy male. Okay, so they produce the XC gamete and X gamete, then here XY. Okay, so you can see XCX. Okay, so she is also a carrier female, and XCY. So here XC means one X is affected. So here the disease is transferred to the sons, right? So that is called as the crisscross inheritance. Sons ಗೆ transfer ಆಯ್ತು ಅಲ್ವಾ, color blind male ಆಯ್ತು. Then XX, she is the normal female, and the last one XY, healthy male. So like this disease transfer from generation to the next generation through the X chromosomes. Hence it is called as the sex-linked chromosome, sex-linked disease. Okay, yes. So these what we studied, these all are the Mendelian disorders. What we studied, sickle cell anemia, thalassemia, phenylketonuria, color blindness, and the color blindness, and another one is hemophilia, right? So here some are autosomal recessive disorders and some are sex-linked recessive disorders. So let us see about the chromosomal disorders. What do you mean by chromosomal disorders? Here, chromosomal disorders means the number of chromosomes that is changed. So the disease caused, that is because of the additional copy of chromosome or a less number of chromosomes. Okay, so that is the chromosomal disorder. So there are two types of the chromosomal disorder: aneuploidy and polyploidy. Okay, aneuploidy and the polyploidy. What do you mean by polyploidy? Polyploidy and euploidy. So there are two, the chromosomal disorder that may, there are two types. Aneuploidy means there is a addition number of chromosomes or less number of chromosomes than the normal chromosomes. ಈಗ 46 chromosomes ಇದೆ, ಇಲ್ಲಿ 45 ಆಗ್ಬಹುದು ಅಥವಾ 47 ಆಗ್ಬಹುದು ಅಲ್ವಾ? So addition or deletion of the number of chromosomes, that is called as the aneuploidy. Polyploidy means, ಕೇಳ್ತಾರೆ ಇದನ್ನ, aneuploidy ಮತ್ತೆ polyploidy ಅಂದ್ರೆ ಏನು ಅಂತ ಕೇಳ್ತಾರೆ. Polyploidy means the total sets of chromosomes. ಈಗ 22 pairs ಇದೆ ಅಲ್ವಾ, ಸಾರಿ 23 pairs of chromosomes are there. So that is the diploid condition ಅಲ್ವಾ. So diploid condition, sometimes this diploid condition that may convert into tetraploid condition, commonly occur in plants. Human beings ಅಥವಾ animals ಅಲ್ಲಿ ಇದು ಕಾಣೋದಿಲ್ಲ. ಡಿಪ್ಲಾಯ್ಡ್ ಕಂಡೀಷನ್ ಇರೋದು ಟೆಟ್ರಾಪ್ಲಾಯ್ಡ್ ಕಂಡೀಷನ್ ಆಗಬಹುದು ಡ್ಯೂ ಟು ದ ಸೆಲ್ ಡಿವಿಷನ್ ಆಫ್ಟರ್ ದಿ ಅನಾಫೇಸ್. ಓಕೆ, ಟಿಲೋಫೇಸ್ ಅಲ್ಲಿ ಏನಾಗೋದಿಲ್ಲ, ಸೆಲ್ ಡಿವಿಷನ್ ಆಗದೆ ಇದ್ರೆ ಏನಾಗ್ಬಿಡತ್ತೆ, ದ ನಂಬರ್ ಆಫ್ ಕ್ರೋಮೋಸೋಮ್ಸ್ ದೇ ರಿಮೇನ್ ಇನ್ ದ ಸೇಮ್ ಸೆಲ್, ಒಂದೇ ಸೆಲ್ ಅಲ್ಲಿ ಇದ್ದು ಬಿಡತ್ತೆ ಅಲ್ವಾ. ಅದಕ್ಕೆ ಏನಾಗತ್ತೆ ಅಂದ್ರೆ ಅಲ್ಲಿ ಪಾಲಿಪ್ಲಾಯ್ಡಿ ಆಗ್ಬಹುದು, ಡಿಪ್ಲಾಯ್ಡ್ ಅಲ್ಲ, ಟ್ರಿಪ್ಲಾಯ್ಡ್ ಇರ್ಬೋದು ಅಥವಾ ಟೆಟ್ರಾಪ್ಲಾಯ್ಡ್ ಇರ್ಬೋದು, ಪೆಂಟಾಪ್ಲಾಯ್ಡ್ ಇರ್ಬೋದು, ಹೆಕ್ಸಾಪ್ಲಾಯ್ಡ್ ಇರ್ಬೋದು, ಈ ತರ ಕಂಡೀಷನ್ ಆಗುತ್ತೆ ಮೇನ್ಲಿ ಇನ್ ದ ಪ್ಲಾಂಟ್ಸ್. ಓಕೆ, here the first chromosomal disorder, that is the Down syndrome, that is example for the aneuploidy. So what do you mean by Down syndrome? Here the cause of this genetic disorder is the presence of an additional copy of the chromosome number in the 21st chromosome, that is the 21st trisomy. It is also called as 21st trisomy. Here this disorder was first described by Langdon Down in the 1866. The affected individual is short statured with broad flat face, wrinkled tongue and partially open mouth. ಡೌನ್ ಸಿಂಡ್ರೋಮ್ ಇರುವಂತಹ ಒಂದು ಪೇಷಂಟ್ಸ್ ಅನ್ನ, ಅಫೆಕ್ಟೆಡ್ ಪೇಷಂಟ್ಸ್ ಅನ್ನ ನೀವು ನೋಡಿರ್ಬಹುದು. ದೇ ಹ್ಯಾವ್ ದ ಶಾರ್ಟ್ ಸ್ಟ್ರೆಚರ್ ವಿತ್ broad flat face, wrinkled tongue and partially open mouth. Palm is broad with characteristic palm crease. Physical and psychomotor and mental development is retarded. Okay, so ಈ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಏನಿದೆ ಅಲ್ವಾ, ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್ ಇದೆ. ಇದನ್ನ ನಿಮ್ಗೆ MCQ ದಲ್ಲಿ ಕೇಳ್ತಾರೆ, ಹಾಗೆ ಬೋರ್ಡ್ ಎಕ್ಸಾಮ್ ಅಲ್ಲೂ ಕೂಡ ಕೇಳ್ತಾರೆ. Here what is this 21st trisomy? 21st chromosome ನೋಡಿ, 21st chromosome ಏನಿದೆ, ಸೊ ಎರಡು ಇರ್ಬೇಕಿತ್ತು ಅಲ್ವಾ, ಈ ತರ ಎರಡು ಇರ್ಬೇಕಿತ್ತು, ಏನಾಗಿದೆ, ಮೂರಾಗಿದೆ ಅಲ್ವಾ. ಸೊ ಟೋಟಲ್ ನಂಬರ್ ಆಫ್ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಎಷ್ಟಾಯ್ತು, 47 ಆಯ್ತು ಅಲ್ವಾ. ಸೊ ಇಟ್ ಈಸ್ ಅ 21st trisomy or it is also called as the Down syndrome. Next one, Klinefelter syndrome. So this syndrome, a genetic disorder caused due to the presence of additional copy of X chromosome in a karyotype, that is 47 chromosomes XXY. So here additional copy of X chromosomes. So total ಏನಾಗ್ತಿತ್ತು 22 autosomes plus XY chromosomes ಇರ್ತಿತ್ತು ಅಲ್ವಾ. ಇಲ್ಲೇನಾಗಿದೆ 22 chromosomes plus XXY. So here total 47. So that is due to the presence of extra X chromosome. Such an individual has overall masculine development, however the feminine development also. XY chromosome, that is the female, sorry, that is the male, right? But here XXY is there, so masculine development, male characters will develop. However, female character is also development of some characters like ovaries and breast. So such individuals are sterile. XY chromosomes, that is additional X chromosome is present. So total number of chromosomes, that is 47. So that is the Klinefelter's syndrome. You can see the, yes, ಇಲ್ಲಿ ನೋಡಬಹುದು ನೋಡಿ, XXY chromosome ಇದೆ ಅಲ್ವಾ. So what are the characteristics of this? That is very important. Next one, Turner syndrome. This disorder is caused due to the absence of one of the X chromosome. So the chromosome is absent totally. 46 chromosomes ಇರಬೇಕಿತ್ತು ಅಲ್ವಾ, 45 chromosome ಇದೆ. So which chromosome is absent? With X0. Okay. Here 45 chromosomes XX illa X0. Okay. Such females are sterile as ovaries are rudimentary besides other feature including lack of other secondary sexual characters. XY ಇರಬೇಕಿತ್ತು or XX ಇರಬೇಕಿತ್ತು ಅಲ್ವಾ. So here one X chromosome is absent. Sorry, not Y. X chromosome is absent. So these are females but with a rudimentary ovaries, means they are not functioning and including the lack of other secondary sexual characters. Okay, so that is the Turner syndrome, very important for your exam. So this completes the chapter principles of inheritance and the variation. Okay, so totally what we learned in this chapter, very important things, I just want to recall that. The first one is inheritance of one gene, Mendel's experiments and why he selected the pea plant for the experiments, that is also important. And inheritance of one gene, that is very important, and some terminologies. And inheritance of two gene, where the law of independent assortment is explained, and here inheritance of one gene, and exceptions like incomplete dominance, that is very important with example, incomplete dominance, then codominance, that is very important. After that, very important about the some diseases, right? Sorry, linkage and recombination, very important. Then sex determination, all organisms. So what we discussed, sex determination, that is also very important, sex determination. Okay, after that, pedigree analysis, pedigree chart, that is also very important. And the last one, disorders, Mendelian disorders as well as chromosomal disorders. Mendelian disorders and the chromosomal disorders, that are very important for your exam. I hope these classes are very helpful for your exam preparation. Okay, if you have any doubt, you can write down in the comment box. I will try to answer for that. So continue in the next chapter. Thank you.